All right, hello students. Here we are in Module 5. This is a new recording, Module 5 in the third edition of the Apologia Biology textbook is quite different from previous editions. In fact, I would probably consider Module 5 to be the most difficult module in the book. There's a lot of technical stuff that goes on here because it is telling us how the cell is actually working and getting everything accomplished. All right, so because cells have their specific jobs to do, and they have to get it done efficiently with enough energy. And so therefore, when we're looking at the running of biological processes, we have to understand it's all about energy. Just like with the economy, everything's about gasoline, right? It's all about oil and gas and energy. So we have the same thing here. How does the cell run? What is it doing? What kind of work is being accomplished? Okay, so we're going to see there's different kinds of work, right? So we have mechanical work, that's going to be the physical part of it, the movement part of it. Think about muscles moving. Transport work, also moving. In fact, if anything's moving, then you know that energy is involved. It has to be. In order for something to move, energy has to be applied to it. And then chemical work is also moving, okay, because we're having chemicals coming together, they're joining, they're taking apart, all this kind of thing in order to get the energy for the mechanical and the transport work. So if we think of the cell kind of like a city, we're going to watch a video here in just a minute. The city has to be run efficiently, right? So in the nucleus, we're going to have, the mayor of the city is going to be telling all of the other groups what to do. And so they have to carry out their work. For instance, we know the mitochondria would be indeed the energy department of the city, right? So let's watch this fun little video to get an idea of that.
If you were an astronaut viewing Earth from outer space, it would be hard to see how human life is organized. You would need a much closer look to see the structures of individual communities. But once you did, you would see that these communities are highly organized. Although it may seem like a big leap to compare a lively city to a living cell, they both have many similarities. All cities need specific jobs performed to make the community run smoothly, and cells are exactly the same. Cells have tiny little structures called organelles that are like workers in a city. Organelles work together to make sure the cell is functioning properly to maintain homeostasis. In this video, you'll learn some analogies that compares the parts of a city to the organelles of a cell. Cytoplasm is like the land that the city is built on. It helps keep the cell shape and the organelles in place. The cell membrane is like a city's border because it controls what goes in and out of the cell. The cell wall is like a police officer because it provides some cells with protection and support. The nucleus is like the mayor's office because it controls the cell's activities. Mitochondria are like power plants because they produce energy for the cell. The chloroplast is like a solar farm because it converts the sun's light energy into energy for plant cells. The nucleolus is like an assembly line because it creates and assembles ribosomes. Ribosomes are like bricks because they make the proteins that are the building blocks of all living structures. The Golgi body is like a post office because it modifies, sorts, and packages proteins. The endoplasmic reticulum is like a roadway because it helps transport proteins. Lysosomes are like garbage trucks because they remove the cell's waste. Vacuoles are like water fountains because they store needed materials, such as water, for the cell. Transport vesicles are like shipping trucks because they move molecules around in a cell. Thank you for watching. Hope you enjoyed the video.
Okay, so I like that little video. I hope you were able to see it. If it gets cut out, I apologize. It just shows how the, cells like a city. Okay, so here what we're looking at is an interesting little graphic, and what's happening here is a muscle is contracting. So this is our mechanical movement, all right? In order for the muscle to move, just if you move your arm, right, a muscle has to move, this extraordinary stuff has to go on. So we have these proteins all along right here called myosin and actin, and the myosin and actin are changed by calcium, and then the myosin heads come in, and then they pull the muscle, and you'll see down here in a second, see how they're going to come together? Does it show them coming together? Not really. It's supposed to show them coming together, which is a contraction of a muscle. Okay, so these are called sarcomeres. If you take biology 2 someday, you'll learn about sarcomeres. It's quite complex, and that's just simply to get a muscle to move. We don't think about it, right? It's all working beautifully because the design that God has put in place is so beautiful. Because when that little green thing moves away, it opens a little portal right here that this purple myosin head can grab a hold of. All right, here we're going to see once again some motion, right? Things are happening. We're moving things. This is transport. All right, so this is the one of the other, one of the other work aspects of a cell. So you can see things are moving. So in order for things to move, we have to have energy. So what are we having here? We're having these little yellow dots go in. Those dots are sodium. And then the potassium, the green ones, are coming into the cell. So sodium out and potassium in. And you'll notice it'll come up here. We're going to have energy to make this door open, and it comes in the form of this right here. ATP, the phosphate drops off, leaving a lot of energy behind, allowing for that little hinge door, if you will, to open. This is called the sodium-potassium pump. It is part of sending an electrical signal down a nerve cell. Also, once again, quite complex. If you take biology 2, you'll learn sort of the mechanism of this, which gets quite complex. All right, we're going to see here some chemistry, some chemical, motion here, chemical work. And what we have is we have a large molecule of sucrose. I think that's how we started. Let's start over here. Yeah, sucrose is a large, what we call a disaccharide. And in here we dehydrate it, or we add water to it, actually. We hydrate it. We break it into 2, and we have glucose and fructose. This thing down here is a chemical complex known as an enzyme, and that enzyme helps to make this process take place. The reason we want sucrose broken down, sucrose, by the way, is table sugar. We want it broken down into glucose and fructose so we can use it better. In fact, glucose is the fuel for running the cellular energy thing. And fructose is very similar. It's just a different shape. It has all the same components, just a different shape. It can be turned into glucose very easily. All right, so the process of taking energy and using it and changing it and making the cellular systems run, that process is called metabolism. If you remember back at the very beginning of the year, we talked about the criteria for life. Metabolism is one of them. Metabolism being the ability to get and use and change energy, okay, to get the cells moving. So we can see right here we have a large molecule right here, and our metabolism is going to break it down, all right. The process, by the way, of breaking larger molecules down is called catabolism, and putting molecules together is called anabolism, okay. And so we have to have energy to do that. It requires energy. All right, energy comes from 2 major processes. All right, we're going to see in your book, look, I believe this page number is right, figure 5.3 on page 181. And these 2 major processes, this is the magic of all life, y'all. This is it. It's all about this. This is the chemistry of life. And those are photosynthesis. We know where that happens. That happens in plants. And respiration. Respiration is what we're doing. In fact, when you hear the word respiration, you might think, oh, that sounds like breathing. That is correct. For human beings, for mammals, respiration is breathing. Breathing is the opposite of photosynthesis. Okay, there are other creatures out there, other life forms out there that have a form of cellular respiration that doesn't involve breathing, but we still call it cellular respiration. Typically, not 100% of the time, but typically it does require oxygen at some point in the process, which you will learn about here momentarily. All right, so it's all about making energy, right? The energy molecule is something called ATP. We saw that in that little graphic with the little hinge door opening up. ATP stands for adenosine triphosphate. Fortunately, I don't think you have to remember that. All you have to remember is ATP. And so when anybody ever asks you how does the cell perform its energy, how does the cell get its energy, you say ATP. And guess where ATP is made? Any guesses? It's made in the mitochondria. That's right, because it's the powerhouse, right? So what is ATP? Well, it's a large molecule. We call it the energy molecule. We have a nitrogen base, all right, called, adenosine, adenine. We have triphosphates. We have the sugar or a ribose aspect and the 3 phosphate groups. So when we transfer energy, the phosphate group is moved between the molecules, and therefore, since the phosphate group, if you remember from the little graphic, the little P dropped off the ATP, that's adenosine triphosphate, it dropped off making adenosine diphosphate, and that one little phosphorus, it showed it attached to that little purple, gateway thing. That's a little simplified, but that's okay. But because it's the phosphate that has the high energy bond that gets released, we call that process phosphorylation. All right, that's what's important about that phosphate. So you're already seeing the importance of things like calcium, and now we have the phosphate groups, phosphorus, and we have our sugars, right? All these chemical compounds that are important for life. So here it is. We're going to have our ATP. The ATP, by the way, is assembled and made in the mitochondria. It's going to go through a process of releasing the energy. You see it says heat released. That's the same thing as saying energy released. We can see here it's adenosine diphosphate with that little phosphate group hanging off, doing the work, opening the door, if you will. And it can go back through by adding energy. See energy coming out, energy going in. Look how the energy goes then through a process called phosphorylation. All right, so we're going to have that happen. This right here is happening in the mitochondria. And it's a cycle that takes place. The ADP can break another phosphate off of there, but typically it'll go back and get recycled to ATP. Let's watch a little video about it.
Almost everything an organism does requires energy. But where does this energy come from? To answer this question, let's zoom in on one of this hummingbird's cells. Notice that each cell contains many mitochondria, which are often referred to as powerhouses of the cell. This is because mitochondria are responsible for producing most of the cell's energy in the form of ATP. ATP, or adenosine triphosphate, is really important because it serves as an energy carrier in cells. It's responsible for moving the energy found in chemical bonds of our foods to the areas of the cell that need energy. Without a constant supply of ATP, cells would quickly die. Let's zoom in to take a closer look at ATP's components. ATP is composed of a nitrogenous base called adenine, bound to the sugar ribose, and 3 phosphate groups. The bonds between the phosphate groups are high energy. An enormous amount of energy is packed into these bonds. When energy is needed, the terminal phosphate group is removed in a process called hydrolysis, releasing energy and forming adenosine diphosphate. The energy released from this reaction is used by the cell to carry out all the functions necessary to sustain life. Later, at the mitochondrial level, ADP and phosphate are recombined back into ATP, recharging the energy pathways of the cell. This cycle, from ATP to ADP and back again to ATP, is called the ATP cycle.
All right. There we go. Yeah. So it kind of simplified things a little bit, which is fine. That's all we need right now in Biology 1. In Biology 2, you'll learn all the details. Oops. There we go. All right. So here's what the molecule looks like somewhat, right? We have our adenine or ribose and our phosphate groups. And as I said in the video, these bonds right here between the phosphates are high energy bonds. Think of it kind of like a spring that if you pull a spring back and hold it, right, and you let go, it boing, it springs out. So that's kind of what's happening with this phosphate group right here. And like I said, we could break this bond as well, right? But typically what happens is we break this bond, we use that energy, and then we take the ADP back into the mitochondria to be recycled into ATP. All right, so in some booklets, you're supposed to sketch this little deal. It's kind of like the picture I showed you earlier. But you have our ATP will perform all the work, cellular work, mechanical work, transport work, chemical work, all the work that is done. It turns into ADP because that phosphate was used, and then it gets energy sources, carbs, lipid, protein, phosphorylation, back to ATP. All that happening in in the cellular level, especially in the mitochondria. Is glucose necessary for the ATP cycle? Absolutely. That's all about glucose. So next time anybody asks you, well, we shouldn't eat sugar. Oh, well, sugar is absolutely the stuff of life, y'all. But it's not the sugar that comes on your table at the restaurant and you pour it into your tea, right? That's sucrose. That's a really big sugar. It's glucose. If I had a bowl of glucose in front of you and you tasted it, it wouldn't taste very sweet. But if I took fructose and you tasted that, it would taste really sweet. So what we do is we put fructose and sucrose together, fructose and glucose together to make sucrose, and it's just right? So that's the sugar we eat in our tea. All right, so supposedly, I would say it's true. You need to memorize the photosynthesis equation just for the rest of your life. Maybe you'll be on Jeopardy someday and you'll need to know this. Okay, but it is a chemical reaction that's happening inside the leaves of plants. We'll see a couple of videos to help explain that a little bit. So what the leaf of a plant needs, first of all plant leaves are green. As we proceed through this year, we'll get into some botany modules, which are so much fun to look at leaves and stuff. So what they're going to need is they're going to need 6 carbon dioxides plus 6 waters. And then the energy, that's where the energy, remember, energy has to come from somewhere. Originally, it's coming from the sun, and it's hitting the chemical inside the green leaf. That chemical is called chlorophyll. And it's making these things. If you did this for chemistry, you would see that this balances out to C6H12O6, that's glucose, the stuff of life, and a leftover that it doesn't even care about is oxygen. What do we care about? Yeah, we want the oxygen, right? We want the oxygen. We breathe that in. We eat food, and therefore we survive. This is why we need to have oxygen, okay? Because it's going to take this, if I turn this arrow the other way, after we eat our salad and breathe, we're going to make carbon dioxide. You're going to breathe it out. And water is going to come out with your breath as well. Okay, so let's talk about chlorophyll. Chlorophyll, like I said, is green. Why is it green? That's what God picked. Not sure why he could have picked anything, but he picked green. All right, but there are various pigments that are found in different leaves. You've probably seen leaves. When I told you every plant is green, you might have argued and said, wait a minute, I have a purple plant outside. You know, what about purple cabbage? That kind of thing. Well, okay, that's a good point. It turns out there are other pigments in there. But guess what? If you take those pigments away, the green is still there. I promise it is. Okay, when we get to the plant section, I'll tell you there is one weird exception about a plant that does it that's not green, but it's very strange. Okay, here we go. What we have is a group of pigments containing chlorophyll A and B. Here's A and B. See how A looks kind of purple? It's up in here, this area right here. And B looks green. And we find that in algae, green plants, and cyanobacteria. In fact, cyanobacteria, also known as blue-green algae, it's actually bacteria. It's necessary for photosynthesis, allowing the absorption of energy from light, okay? So that's what our chlorophyll is doing. It's just the chemical compound that performs the work. Does life on Earth depend on the process of photosynthesis? What if you don't eat any plants? Are you a carnivore diet person? Interesting. But guess what? We absolutely have to have plants, because what did that cow that you're eating for lunch have for breakfast? Grass. Okay, so they needed the green stuff. And so the energy ultimately comes from sunlight. It is transferred into the leaves, and the leaves will store it in various places. Either it'll store it in the fruit that comes from a flower, in the root, like for instance a carrot. Sometimes the stem, there are some stems we eat like asparagus, celery, you know, the energy stored there. And we get energy from the plant. Even if we don't eat plants, something we ate got energy from it. All right, let's watch a little video.
Light gives plants the energy they need to grow. But what happens if plants receive too much light? The answer is they glow. This neon glow is actually happening around us all the time. The result of a cellular process where light from the sun is transformed and released as fluorescent light. A journey into the plant cell reveals how it works. This is a chloroplast, the energy-producing organ found inside the cells of plants. When sunlight strikes a plant, disc-like structures within the chloroplast absorb the light and convert it into energy. However, a small fraction of this light, about 1%, is emitted as fluorescent light. The light exits the plant cell and is released into the atmosphere. The amount of light released can vary depending on factors like the time of day, time of year, and how much sunlight is being absorbed.
Humans can't see this light. Our eyes just aren't that sensitive. To see the fluorescent light that's emitted from plants all over the world, we have to use scientific instruments that are placed on satellites.
Earth observation satellites, outfitted with special sensors, are able to detect this light from space. After multiple orbits, scientists can construct a detailed view of the data. This is what plant fluorescence looks like on a global scale. The visualization was created from 5 years of satellite measurements, analyzed by a team of researchers led by scientists at NASA's Goddard Space Flight Center. Each point of light on this globe represents fluorescent light exiting a plant cell. The greater the intensity of light, the brighter the color. By observing changes in intensity over time, scientists can distinguish stressed, dead, or dormant plants from healthy and growing vegetation.
Okay. I think I stopped there because that's the part of that we needed. I hope that came through and y'all were able to watch that. It talked about how really leaves are kind of glowing. We can't really see them without special equipment to see it. All right, so the next question we have is, what two products of photosynthesis are essential for life on the earth? Okay, what are the products? Right here, y'all. Right here, glucose and oxygen. Okay, so sunlight, carbon dioxide, water going in, oxygen, glucose. Interestingly, you notice this plant doesn't talk anything about the soil that the plant needs, you know. So think about the soil. I don't know if you've ever heard of plants that just grow in water, right? Hydroponics. If you think about the soil like as vitamins, it also holds them in place. So it's important that way. But they don't really get much from the soil except they get stronger from vitamins and minerals, you know, that come from the ground. But to do the actual process of making the oxygen and glucose, all they need is sunlight and water and carbon dioxide. All right, so on page 195, I think that page number is correct. You're going to see that we're going to be looking at the electromagnetic spectrum. Why are we doing that? Why are we looking at that? Well, for one thing, I showed you how those leaves glow, and so we're going to see that. So it turns out that when we look at energy, we're talking about energy. The energy is contained, if you will, in this broad thing called the electromagnetic spectrum, or the EM spectrum. It's kind of crazy because everything fits into there. It's like God has tied everything into this beautiful little package and wrapped it with a bow. All right, so the visible light we're familiar with, right? Roy G. Biv, red, orange, yellow, green, blue, indigo, violet. Those are the things we can see. Beyond visible light, notice what a small part of the spectrum it is. Beyond that, we have ultraviolet. Behind it, we have infrared. We can't see any more here, and we can't see any more here unless we have special equipment. And then as the energy increases, look how these waves are getting closer and closer together. That means they're higher and higher energy. All right, up to the point of the gamma rays. The gamma rays are used, they can actually kill cells. I don't know if you've ever had an x-ray taken, right? It goes right through your skin. It shows your bones and other hard things that are in there. On the other side, when we get less and less energy, notice we have microwaves. That's the same, you know, it's in your kitchen probably. We have radio waves, they're all around us. And then we're going to see over here, and it turns out probably somewhere in here, I'm not sure where, it's kind of still being, you know, discovered at this time, is that gravity somehow fits into this as well. It's crazy stuff, crazy. You have a lab in your book called The Pigments of Photosynthesis. And so it's on pages 186 and 187. This is a good one to do. It's fun to do. I can't remember exactly what the procedure was, but in my classes I would normally take a leaf, like a spinach leaf. Spinach works really well because they're kind of soft, and you sort of wrap a quarter around it so it's rough, and you put it on a piece of filter paper. Coffee filter paper would work, even a paper towel would work, and let water just pull it up. You could also maybe try to use alcohol to see if that does a better job. That's a fun little experiment. It says to remember something, I don't know. But we're looking at more pigments to consider. Chlorophyll is not the only pigment. It happens to be green. But there are other pigments that are found in plant cells. Not really in animal cells, okay, but in plant cells, we're going to see these things called carotenoids. You hear like the word carrot, right? So those are going to be the oranges and yellows. We're going to see that in pumpkins, oranges, bananas, things like that. The anthocyanins, one of my favorite, the reds and the purples. It turns out that if you can find foods that are naturally rich in anthocyanin and phycobilins, here's the phycobilin, blues and reds. If you find naturally blue, red, and purple things like blueberries, purple cabbage, eggplant, those have these wonderful chemicals, these anthocyanins, phycobilins, that are actually very good for us. So it's really good to eat the dark things. They're talking about eating the rainbow kind of a thing. But you may have to write these things down, right? Just remember carotenoids, orange and yellow, anthocyanin, red and purple, and phycobilins, blue and red. Okay, so that gets us to, I love this time of year, although in Texas we don't see much of the fall color, if you will. We do see some change. In Texas, mostly we go from green to brown, but that's okay because the brown is also something in there. It's a type of acid called tannic acid. It's the same thing that we use to make tea. That's why tea is brown, right? Because it comes from leaves of the tea plant, and those leaves have generally, well, we call it black tea because they let them dry out, Green tea is when they're still green, when the leaf is still green. But it turns out, so if I have green here, the chlorophyll is being shown. Well, if the green gets taken away, and how would that get taken away? Well, in the fall, guess what? The plant cuts off the water supply to the leaf. Have you ever wondered why the leaves fall off of most trees? Not all of them, but most. It's because the water gets cut off. And there, when the water's cut off, guess what? Photosynthesis can't take place. So the green just sort of dies away or just fades away. And then the other colors are then visible. That's why in the fall we see the yellows, the reds, the oranges, the browns, right? Because the green is gone. That's why we see it. And then they fall off. Why do they fall off? Because they don't have any more water coming to them. They don't have any more photosynthesis. They're going to fall off because they're going to die, and the plant will go through the winter and conserve all of its energy and the rest of the plant, and the next spring will send out some fresh green leaves in most cases. All right, let's watch a video.
The leaves of this plant have cells that carry out photosynthesis. During photosynthesis, plants capture the energy from sunlight and use it, along with water and carbon dioxide, to produce carbohydrates, or sugars. Oxygen is also released as a byproduct. These photosynthetic reactions are typically written as a chemical formula. Solar energy plus 6 carbon dioxide molecules plus 6 water molecules react to produce 1 glucose molecule and 6 molecules of oxygen. To better understand the reactions of photosynthesis, let's take a closer look at the leaves of this plant. If we zoom in on one photosynthetic plant cell, we can see the chloroplast, where the reactions of photosynthesis occur. Let's zoom into this chloroplast to get a closer look. Photosynthesis consists of 2 primary steps, the light reactions and the Kelvin cycle reactions. Both steps occur within the chloroplast. The light reactions occur within the thylakoid of the chloroplast. Here, special pigments absorb light energy and transfer it to high-energy electrons, which ultimately produce ATP and the electron carrier NADPH. Water is also split to replace the high-energy electrons. In the process, oxygen gas is released. The Kelvin cycle reactions, commonly called the dark reactions or the light-independent reactions, occur within the stroma of the chloroplast. Here, a series of reactions use the ATP and NADPH produced by the light reactions to turn carbon dioxide into carbohydrates. The NADP+ and ADP that were also produced by the Kelvin cycle reactions are then recycled to be used by the next set of light reactions. Now that you understand a little about light reactions and Kelvin cycle reactions, let's zoom back out and do a quick review. During photosynthesis, plants capture the energy from sunlight and use it, along with water and carbon dioxide, to produce carbohydrates, or sugars. Oxygen is also released as a byproduct. These photosynthetic reactions are typically written as a chemical formula. Solar energy plus 6 carbon dioxide molecules plus 6 water molecules react to produce 1 glucose molecule and 6 molecules of oxygen.
Okay. All right. Did that give you a little bit of a headache? Yeah, it's complex, y'all. It's it's simple, right? Because that's a very simple balanced chemical equation, but the complexity that goes into that just shows you, God's kind of showing off there. Yeah, it's pretty cool stuff. All right, so we're going to be looking now, apparently in your book on pages 188 and 89, we're going to see the different parts of the plant cell. Okay, so this is kind of fun. In fact, if you have a leaf nearby, especially like I said, the spinach, if you think about it, if you squeeze it between your fingers, you'll feel it's a little bit spongy. It's pretty thin, so you may not be able to tell very well. Some of them, thicker leaves, if you squish them, all right, so try that. Go outside and find some leaves and squish them and see how they're a little bit spongy. So it turns out, and also take a look at the top. Generally, the top of the leaf is more green than the bottom of the leaf. It's greener. It's a darker or a brighter green. The reason is because all of those cells are packed in there. Why do you think they're packed in there? When they're facing the sun, right, the sun's going to get those right there quickest. We're going to see there's other stuff. Notice it's called the spongy mesophyll. This is palisade. Palisade means all lined up like a brick wall, if you will, right there. And then we have the spongy because there's spaces in between. When you squish, it's literally air that you're squishing out of there. Here we have some of the transport type of cells called xylem and phloem that are inside there. This is a fun deal right here. This is called a stoma. It is a hole. Stoma means hole. And there are cells that they allow. This is where the oxygen comes out and the carbon dioxide goes in. Beautiful, beautiful design right there. And sometimes these are crunchier than other times. If they have more of these, they're crunchier like celery. And sometimes not as much. So I'm not sure if you're supposed to draw this, but just like I said, take a look at a couple of leaves. Maybe if you have some house plants or something like that, take a look at those. All right, so here we go. Our photosynthesis, like in our video that showed the complexity of it. All right, we're going to have the sunshine right here coming in. For our light-dependent reactions, we look at our ATPs being made. We have these other chemicals. These right here are called hydrogen thieves or stealers. They have to get that hydrogen and carry it around. When you get to biology 2, you'll have to know those things. Calvin cycle, also known as the Krebs cycle. And this is where we're going to break everything down to end up with all those carbons off of the sugar, from the carbon dioxide into the sugar called glucose. All right, so once again, very complex. Calvin cycle, also known as Krebs. Chloroplast, the chloroplast looks like this whole thing right here, a little bit like a, jelly bean, and inside we have these stacks of pancakes, green pancakes. And that's where the chlorophyll is actually in these thylakoids right here. I'm sorry, I always say that wrong. The thylakoids right here. So we have the thylakoids with the green chlorophyll in there, forming into a group called the chloroplast. I'm not sure if you're supposed to draw this one as well. I believe this might be another complex but very useful video. Okay, but so just listen closely. If you just hear it, that's good. You don't have to know all the details, but just listen to it.
Photosynthesis consists of two primary steps, the light reactions and the Calvin cycle reactions. In this tutorial, we'll focus exclusively on the light reactions. The light reactions occur within the thylakoid of the chloroplast. Here, special pigments absorb light energy and transfer it to high-energy electrons, eventually producing ATP and the electron carrier NADPH. Let's zoom into the thylakoids to take a closer look at how ATP is created. The light reactions use two photosystems, called photosystem 1 and photosystem 2, which are both embedded in the thylakoid membrane. It's important to realize that these photosystems are named for the order in which they were discovered, not for the order in which they participate in the photosynthetic process. The light reactions actually begin at photosystem 2. The first thing that happens is that photosystem 2 receives photons, or light energy. This light energy is transferred to a chlorophyll reaction center, causing electrons in the reaction center to become energized. These electrons become so energized that they escape photosystem 2 and move to a nearby electron acceptor molecule located in the electron transport chain. Meanwhile, to replace the electrons leaving photosystem 2, water is split, releasing oxygen, 2 hydrogen ions, and 2 electrons. The first set of electrons continues to move down the electron transport chain, releasing stored energy as it moves. This energy is used to create a hydrogen ion gradient. A protein in the to the low concentration of ions in the stroma. This gradient contains a large amount of potential energy, which is used by an enzyme called ATP synthase. The hydrogen ions flow down the concentration gradient through a channel in ATP synthase, releasing energy in the process. ATP synthase uses this energy to add a phosphate to ADP, forming ATP. Let's zoom back out for a moment and return to our chloroplast. Remember from the beginning of this tutorial that the light reactions produce both ATP and NADPH. We've just seen how ATP is produced, but what about NADPH? Let's zoom back in to take a closer look. Notice these electrons over here where we left them at photosystem 1. As photosystem 1 absorbs additional light energy, the electrons again become energized, escaping photosystem 1 and moving down the second electron transport chain. Electrons from the electron transport chain adjacent to photosystem 2 replace those in photosystem 1. And again, water is split to replace the electrons that have moved from photosystem 2. At the end of this electron transport chain, the energized electrons in a hydrogen molecule are used to reduce NADP to NADPH. Let's zoom back out to review. The light reactions use light energy and water to produce ATP and NADPH. Oxygen gas is released as a byproduct. Together, the ATP and NADPH formed during the light reactions are used by the Calvin cycle reactions, which are discussed more in depth in a separate tutorial.
Okay, if you didn't have a headache already, then you probably have one now. So that is a lot of stuff, right? And what fascinates me is that we've kind of figured this out, right? We think this is what's going on. Remember, all science is something that can be questioned. We're pretty sure this is what's happening. We could be wrong about a few of the steps or whatever, but you have to see there's this complexity to this chemical reaction taking place. Typically, I would say the Calvin cycle, known as the Krebs cycle, it's like dun, dun, dun. It's one of the most, frightening things for undergrads to deal with. And so we're going to actually study that next. So if I told you that this part, the light-dependent reaction, was the easy part, you might roll your eyes just a little bit. And remember, you don't have to know all these steps. I'm going to give you a little, like a chart here in a minute for the things that to wrap it up for you to see what you need to know. But I would like you to understand the complexity of this. So let's go now to the Calvin cycle.
Photosynthesis consists of two primary steps: the light reactions and the Calvin cycle reactions. In this tutorial, we'll focus exclusively on the Calvin cycle reactions. The overall purpose of the Calvin cycle is to convert carbon dioxide from the atmosphere into carbohydrates, or sugars, which the plant needs to power its cellular activities and build new plant structures. The Calvin cycle reactions occur within the stroma of the chloroplast. The reactions use the ATP and NADPH produced by the light reactions to convert the CO2 to carbohydrate. Let's zoom into the Calvin cycle to take a closer look. The Calvin cycle could be divided into three phases: carbon dioxide fixation, carbon dioxide reduction, and regeneration of RUBP. The first phase, carbon dioxide fixation, basically captures the CO2 from the atmosphere so it can be used in the reactions. To do this, CO2 is attached to RUBP, a 5-carbon molecule. The enzyme used in this reaction is called rubisco, and the result of the reaction is an unstable 6-carbon molecule that quickly splits into two 3-carbon molecules called 3-phosphoglycerate, or 3PG. Next, the process of converting the CO2 to carbohydrate begins. This phase is called the carbon dioxide reduction phase because we're adding electrons and energy to the CO2 molecule. During this phase, a sequence of reactions uses NADPH and some of the ATP from the light reactions. These molecules supply the needed electrons and energy for CO2 reduction. Electrons are added from NADPH, and through a series of reactions, 3PG is reduced to form G3P, a carbohydrate. ADP and NADP+ return to the thylakoids to be converted back to ATP and NADPH by the light reactions. One of the G3P molecules is set aside to be used as a building block for glucose, but the majority of the G3P molecules move forward into the third phase of the Calvin cycle. In this phase, ATP is used to combine the rest of the G3P molecules to form RUBP molecules. This RUBP can then combine with additional carbon dioxide molecules, continuing the carbon reactions. To form a glucose molecule, the cycle actually has to turn 6 times, because each turn of the cycle adds only one carbon atom from the incoming carbon dioxide. It's important to remember that the Calvin cycle is dependent on the light reactions to provide NADPH and ATP, which in turn provide the electrons and energy needed to form carbohydrates.
All right, there we go. Okay, so I'm going to bring up this chart right now. First of all, I want to apologize. I did misstate something before when I talked about the cycle there. You know, that I said it's the Krebs cycle. It's really not officially called the Krebs cycle. It's called the Calvin cycle. That's because that's what it's called in plants. Okay, in plants it's called the Calvin cycle, and it's somewhat similar to what happens in respiration in animal cells. That's called the Krebs cycle. Okay, so I'm sorry for misspeaking there. In animals it's called the citric acid cycle, known as the Krebs. In plants, it's rarely referred to as Krebs here. It's referred to as the Calvin cycle. So, not that that's important. Just remember, next week when we do 5B, we'll officially be looking at Krebs. And Krebs is the thing that makes us all nervous. But it's just as complex in plants as well. It's kind of the opposite direction, however. Okay, so here is a table, kind of a summary of the steps. Supposedly 1, 2, 3, 4, 5, 6, 7, 8 steps is what we're saying there are here. And so you have our stages of light reactions. We have this thing called the electron transport chain. That's in that first video where the little electrons were going and moving. And if something's moving, you know we have energy there. Okay, and it was going to reattach that electron to that positively charged proton called hydrogen, right? So it's all about the hydrogens there. And the second stage would be that Calvin cycle, or the light independent part. That's the part that's going to go through the circle there. That's going to break down that CO2. Okay, so then we have our input is sunlight and water. Our input is CO2, ADP, and NADPH from here, right? Our output is going to be our oxygen that humans can breathe right there. And then our output on this one is putting all those into a different form to make our C6H12O6. Where is it happening? The thylakoid membrane, that's the little green pancake thing. That's where the light reactions are taking place. And the stroma is just, if you noticed on some of those cells, it's just kind of sort of the middle of the cell. It's happening outside of the thylakoid, if you will, inside of the chloroplast, which is inside, which has chlorophyll inside of it, right? And so that's where it's all taking place, okay? So then we're going to see this is happening there in those chloroplasts. And next week when we look at it, we're going to see everything's going to be taking this stuff and happening inside the mitochondria. Okay, so it's not that the plants don't have mitochondria, they do, but they use the mitochondria to feed them when there's no light, when there's no sunlight, all right? Because they're only doing this right here, this photosynthesis, when there's sunlight available. And at nighttime, they don't do it, right? So they have to make food for themselves. That's what plants are making food for themselves. Okay, so glucose is the main source of metabolic energy in living things. It serves as the main substrate in glycolysis. So we just made it. We just made it in the plants right here, right? We just made that. Here it is. The final product is glucose. That glucose now, next week, we're going to find out, is taken and used in the process called the Krebs cycle, right? Photosynthesis. Okay, it says here, I'm not sure what this video is. Let's check it out.
Earth, the blue planet, dominated by the world's oceans. With the help of microscopic phytoplankton, the Earth's oceans can draw down vast amounts of carbon dioxide through photosynthesis, dwarfing the levels of CO2 stored both on land and in the atmosphere. Australian innovation company Ocean Nourishment Corporation have pioneered a patented carbon removal technique that enhances this biological pump with a low opportunity cost and global scale potential. The process works by boosting nutrient-deficient areas, or so-called ocean deserts, enhancing phytoplankton's ability to absorb and export carbon to the deep ocean. ONC will use state-of-the-art vessels to deliver its ocean plant food formula, including green ammonia, derived from renewable sources, to boost phytoplankton growth and enhance atmospheric CO2 removal. The ocean nourishment sites are carefully selected, avoiding protected areas and sensitive habitats. new phytoplankton growth is safely engineered to only 10% of levels seen in natural nutrient upwellings. ONC's carbon methodologies estimate the carbon exported by sinking phytoplankton, and this can be validated using satellite technology and direct measurements. Compared to land-based alternatives, ocean nourishment offers rapid, long-term removal of atmospheric CO2. This will help achieve climate stabilization targets, whilst also sustaining the base of the marine food web, supporting fisheries and other crucial ecosystem services. Join ONC and support our Ocean Nourishment Scale-Up program to help stabilize Earth's climate and revive our oceans.
Okay, I think the reason I wanted to put that in there was, everybody always talks about how, the forests, the rainforests, we're cutting down our oxygen producers. It's true. We do the trees produce a lot of oxygen, but not the majority of it. The vast majority of the oxygen we need for life is being produced in the oceans. Okay, so it's ocean photosynthesis that's producing, I think, 80 to 85% of the oxygen we use. So honestly, what we need to do, not that we shouldn't cut trees down. I don't want to cut trees down either. We do need natural resources like wood, etc. But we need to take care of our oceans, is the truth, because the oceans are really sustaining life. Okay, I apologize for the length of this lecture. There was so much stuff to cover, and I really felt like the videos would enhance your understanding of this. So you may have to split it up into a couple of sessions for listening to. I believe part B will not be as long next time, although it's just as complex because it's talking about the other side, the Krebs side of it, where we use cellular respiration. All right, so read through it, do your study guide, and I hope you understand. I hope you get to do a fun lab. That chromatography lab is a ton of fun. You can do it at home very easily. So just read it in your book and try that. Okay, thank you, students. So long. See you next time.